Hello everyone. Welcome to latest update program. The topic of discussion is hybrid microscope for protein imaging. A team led by Indian scientists at the Marine Biological Laboratory, MBL, has developed an innovative hybrid microscope capable of simultaneously imaging the 3D orientation and position of molecules, such as proteins inside cells. The tool combines polarized fluorescence technology with a dual-view light sheet microscope, DISPIM, allowing for unprecedented clarity in capturing molecular interactions. This breakthrough will help understand how proteins interact with other molecules to perform vital functions in the body. Future improvements will enable real-time observation of molecular interactions in living organisms, advancing our understanding of cellular processes. Drishti IAS in Noida is offering an IAS GS Foundation, prelims plus mains, course in an offline batch. Admissions are open for the English and Hindi medium batch. For more details, contact 87501875501. Now let's discuss about key features. The microscope enables the recording of 3D protein orientation changes, essential for studying protein dynamics and their interactions in living cells. It combines two advanced technologies. Polarized fluorescence enables better control and visualization of molecular orientation. Dual-view light sheet microscope, DISPIM, offers a clearer and more comprehensive view of molecular structures and movements in 3D. The microscope's capability to image molecules from multiple perspectives and record precise orientation changes is crucial in studying dynamic cellular processes like protein folding and interactions. Now moving on to applications and implications. 3. Deep protein orientation imaging can help overcome challenges like imaging molecules in the spindle of a dividing cell, which is difficult to observe at a single angle. This technology offers detailed insights into the way proteins perform their function, which can impact research in cellular biology, drug discovery, and biomedical sciences. It has potential applications in understanding diseases where protein misfolding or improper interactions play a role, such as in neurodegenerative diseases or cancer. Now is the time for practice question. What biological structures will the new microscope help researchers better understand? Option A, mitochondria. Option B, proteins and their interactions. Option C, membrane lipids. Or option D, microtubules. Send the answer for this question in the comment section. Thanks for watching.
For more informative content, like, share and subscribe, and do not forget to press the bell icon to get the notifications.